So the last speaker of this session is Professor Frederick Tam, who is a consultant nephrologist at Imperial College. Fred is going to give us an update on his clinical trial testing the drug fostamatinib in IgA patients. Thank you.
Thank you for the invitation today. I think I see quite a number of familiar faces around. Some of them are the patients or collaborators I've met over a previous meeting. And some of them are the patients I see in the clinic as well. So it's wonderful opportunity to share with you. Now, see if we can put on the slides there. Yeah, this one, yeah. Okay. Great. Yeah. Great. Okay, so we're slides there. So I've been involved in quite a few of these meetings over the last few years. It's quite exciting to give you an update of what we have been doing. Now, so what I'm going to summarize to you is that some of the research work we've done with a lot of collaboration with the patients and also different hospitals, both in UK and around the world. And the main thing to all of you already know the importance of kidney disease. The main story I want to bring to inflammations, because this is the center focus of IgA nephropathy. Now, I've been working in a few miles away from here for many years. On one of the rare days, I managed to capture the blue sky there. And the most important thing to show you here is that this is the main entrance of our hospital, Hammersmith Hospital. What I try not to picture is the Wormwood Scrub a few yards away from there. And my boss always tells me if I do anything wrong, I'll go the other way. And this is our renal unit. This is a clinical center formed by amalgamation of 3 NHS centers about 13, 14 years ago, St. Mary, Hammersmith, and Charing Cross. And this is important because this is what our basis of all our research work is all focused on patients. And this is our laboratory. The good news is that all these are within 5 minutes walk from each other, because a lot of the challenge of kidney research is that the hospital is so remote away from the research center, you can never link things up in a meaningful way. And one of the concerns in IgA nephropathy is that we know IgA is contributing to inflammation and scarring, resulting in kidney failure. And if hypertension is not being controlled, it makes the whole thing worse. But other than that, we actually know very little about step-by-step deterioration of the kidney. And one of the problems is that early, as Jonathan may have mentioned earlier on to you today, early stage of disease is not always clear-cut. And then although we got lots of therapy around in kidney medicine over the years, and some of you may have come to a kidney clinic, you know that there are medications around. So what's the problem? The problem is that some of the medications are very generic, so they're being used to treat a wide range of conditions. Sometimes they work, sometimes they don't. And even when they work, if they're more generic or immunosuppressive therapy, they tend to increase the risk of infection and other complications like diabetes. So what we think of is that we need to do something a little bit more. Hopefully we develop something more effective in a reliable way, and hopefully less side effect. But of course, all these are our intention. The question is it true or not? I think that's where research comes in. Now, so all of you are aware that how it's been diagnosed in the screening stage about urine dipstick test, but the most important thing is kidney biopsy. The reason I bring up the kidney biopsy here is that these green color, they are the antibody IgA got stuck to the kidney of patients, causing inflammation and fibrosis. And this is why IgA nephropathy is being named as such about 50 years ago. And the question is, what happened after that? Now, this is where our limitation understanding is. Now, as a researcher, as a clinician, all of us only know a tiny bit of the whole story. And we are quite blind to what the overall picture is. And my personal understanding is that a lot of my research have been to how inflammatory cell would penetrate the kidney and causing tissue damage. And just bear in mind that don't believe anything I tell you superficially, because you'll hear lots of researchers like Danny, who's interested in genetics. They have a different angle of how to understand the disease. And all these different angle understanding is essential, because if we sort of being blinker and one explanation of everything, like my bias of inflammation, we would never get the full understanding of that. Now, so one of the molecules I've been working on is spleen tyrosine kinase. Now, some of people ask me, why do you work on this molecule? When inflammation is such a big story, you can't say one molecule explain everything. And that is absolutely right. And I think when at the end of the story, I tell you, just bear in mind that there are many other targets we need to think about in the bigger picture, rather than focus on one molecule. And the second thing is that, actually, regretfully, when I look at some of my older paperwork, my answer is, why didn't I study it earlier? Because 2 years before I started this project, I was having open collaboration discussion with a lot of other scientists. There are a couple of people telling me, write this down on your piece of paper, spleen tyrosine kinase, this is important. And what did I do to a piece of paper? I put that away. And it's only many years later I discovered that piece of paper was there. And by that time, I eventually have started working on this molecule. Now, the importance of this molecule is that the honest truth is that we don't know whether it's important in IgA nephropathy. What people know is that spleen tyrosine kinase is an important enzyme in our body. All of us got it. This is important in our defense system. That is what helps our lymphocyte to make antibody response to bacteria and viruses. That is involved in controlling inflammation, how we fight pathogens. So there are several functions that are known to be in our immunological system. But what has not been known is whether it's anything to do with IgA nephropathy. And why didn't we know about that? Partly is a typical scientist is that I'm scared to find out why it's involved or not. I remember a few years ago, or probably getting near 6 or 7 years ago, one of the visiting researchers in my lab, and she told me the project I gave her is really too boring. So I said, okay, what do you want to do? And she said, okay, I want to work on IgA nephropathy. So I asked her, check my literature about IgA nephropathy. So she's going to hope that she'll find all the expert opinion I have. And she came back very quickly within an hour, say, I couldn't find anything among your publication list about IgA nephropathy. So that's the answer. I say, I never researched on IgA nephropathy because it's such a difficult subject. So she pushed me to give her something more interesting. So we speculate that spleen tyrosine kinase being important in the immune system, is there are remote possibilities involved in IgA nephropathy? And we put forward an idea. I would say that it's a speculated hypothesis that the complex of IgA in the patients, somehow they would get stuck to the receptor or surface of kidney cell, in particular mesenteric cells in the kidney. And following that, they would stimulate the spleen tyrosine kinase and to cause release of inflammatory mediators. Now, at that time, we have no evidence whether it's true or not. So we've done some experiments. And some of you have seen that before over the last couple of years. So the first evidence we have is that we, together with Jonathan Barrett and his colleagues in Leicester, we get hold of the serum sample from patients in the freezer, or the serum in the freezer, the patient in his clinic. And Jonathan and his team kindly purified the IgA from them. And then we use it, we've done the easy bit. So we bring it down to London and put it in our cell culture in the laboratory. And then we find that we confirm what other people know is that if you put the patient's IgA in kidney cells in culture, particularly mesenteric cells, they release a whole house of inflammatory mediators. And they are sufficient to cause further kidney damage in a real-life situation. Now, this is not rocket science. People have done that before. All we did is ask someone expertise like in Leicester to help us, and we start the experiment. So the real test is that what if you try to look in the kidney cells of what are the underlying mechanism. And this is a simplified graph of what we did. So we look at the protein expression of spleen tyrosine kinase over time. Now, if you look at time scales very fast, over 30 minutes, then you can see as soon as you put the IgA in the kidney cells, they start to increase the amount of spleen tyrosine kinase, the activated form. For spleen tyrosine kinase to be active, they need to be phosphorylated, which is adding a phosphate group to them. So this is the first piece of evidence we have that spleen tyrosine kinase may have something to do with IgA in patients. And this is work done by the researcher at that time, Minjung Kim. She was the one telling me my research is really boring. So that's how we get started. Now the question is, if we demonstrate the enzyme is there, what is the consequence of blocking that? And can we demonstrate that can reduce inflammation? So we got a collaboration with the pharmaceutical industry at that time. They provide this very useful research tool, which is an inhibitor to spleen tyrosine kinase. And that would block the active site of the enzyme and hopefully to block the inflammation. So we done the experiment at that time. We put this inhibitor in the cell culture of the kidney cells I mentioned to you earlier. We predicted, we don't know the answer, that it would block the production of inflammatory mediators. And that's what we find. So we find here that if we go back to Danny's story about genetics, the next thing below the gene is the gene is affecting production of messenger RNA protein. In here, we look at the messenger RNA of one of the inflammatory mediators. We look at the protein productions. So it's a complex graph, but if I give you a key message is that in the patients, in the presence of IgA from patients, we have production of the messenger RNA protein for this inflammatory mediator up here. As soon as we put increasing amount of this Syk inhibitor in the cell culture media, we can reduce the amount of productions. So this is the first hint that not only we can find this enzyme as a drug target in the kidney cell in culture when we stimulate patient IgA, when we use a pharmacological inhibitor, we can reduce the production of inflammatory mediators. Now, a lot of you probably have a lot of background knowledge about science and inflammation. We all know that one mediator doesn't explain everything. In our body, when we get ill of something, there's a whole family of inflammatory mediators involved. So we screen for a bigger story, and what we find is that the experiment we did, we reduced the production of a full house of cytokines, about 6 different ones together. They got inhibited in the same experiment by this pharmacological inhibitor of spleen tyrosine kinase. Now, there's a problem here. A lot of you know that when you take a medication, you think the medication is doing one thing. They can be doing 5 or 6 different things. They can either be off-target effect because the drug is not selective enough, or it can be a side effect of something that you may or may not desire. So when we got results like I showed you earlier on, we're a bit skeptical. We repeat the experiment about 3 or 4 times. We get the same results every time. But if we just get a gut feeling, it's just too good to be true. It can be something else going on. So we thought we've got to change the approach. We use the same cell culture. This time, we didn't use the inhibitor from the pharmaceutical company. We use a molecular method of silencing the gene expression. And that's what we did in here. So we got silencing RNA that would neutralize any gene expression of spleen tyrosine kinase. And we find out what happened to the cytokine production. And what we find is that we confirm the results we got earlier on. The whole panel of these 6 different inflammatory mediators, they were also silenced by using the molecular method, confirming that pharmacological inhibitor and the molecular method show that spleen tyrosine kinase is a target. Now the question is, all that thing we talk about is the nearest thing to patients is the serum sample from Leicester IgA patients. Other than that, we have no other clue whether it's true or not in real patients. So then that's upon we go back to the kidney biopsy of patients in our renal surfaces. So this is a big archive of sample over time. And one of my PhD students, Steve McAdoo, he done a wonderful job. He sort of started to use immunostaining of this enzyme in the biopsy of patient. All this brown color staining had the spleen tyrosine kinase. And this is negative control with no staining. And what you find is that in real life, every patient is different. And what he found here is very interesting is that when he compared different severity of IgA nephropathy based on the features of a kidney biopsy, of how inflamed they are, the more inflamed the kidney is, the more of the spleen tyrosine kinase expression. The quiet one, the less inflamed one, you can hardly detect any of them. Now that is interesting because we got to face a real world that you cannot expect that every patient respond in the same way. So you got to identify the patient who are likely to benefit by a certain new treatment. So that's our understanding at that time that in future, if this is ever going to be useful treatment, it is the patient who got the more inflamed looking histology of the biopsy. They are the one that we ought to think about as a new treatment. And then what is interesting is that in a wider context is that IgA nephropathy is not the only inflammatory kidney condition involving spleen tyrosine kinase. And we have looked at other conditions like a rare condition called Goodpasture disease, or a medium condition like vasculitis or lupus nephritis. All of them share this mechanism on the renal biopsy there. And what is interesting but also concerning is that when you look at all these patients at the time of biopsy, the one who have more severe renal impairment, like the raised creatinine, they are the one who express a larger amount of this enzyme abnormally in the biopsy. So circumstantially, we have find that in severe disease, you can detect the expression of this inflammatory molecule enzyme, spleen tyrosine kinase. And then they go with severity inflammation. And the question is, where do we go from there? And quite thankfully, I done something which I didn't think very much at that time. One of the visiting researchers from Australia, he came to visit our lab at that time before we published our first paper. He asked me a very simple question. He said, what are you doing these days? And I know in a few months' time, he's going to see all this data we're publishing. So I told him right away what we're doing, show him the data. And he done something really wonderful. So he go home, back to Australia, and find that very interesting. He tried to repeat every single experiment we haven't published yet. And so over the stretch of 5 or 6 years after that, every paper we published, he published a parallel paper about 6 months later, confirming every single piece of data we have. And that has been very useful because one of the concerns about medical research is that not only you cannot trust a piece of paper, sometimes I don't even trust my own data. But if you got different scientists or different doctors somewhere in the world confirming your finding, you know that it is worthwhile to carry on. So to summarize where we were at that time a few years ago, we've shown that spleen tyrosine kinase is involved in kidney cell stimulation by patients' IgA. And blocking that using different approach would reduce the inflammation in the laboratory. And what is encouraging is that in patients' biopsy, you get increased amount of this molecule. So that's when we were very keen to do a clinical trial. And after we back in the industry for a few years, for some unknown reason, they agreed to start the trial. And I was very concerning. They would give me a letter say that I'll support your trial and help you. And they never gave me the letter because I can't write a grant to get the funding for it. And then they told me the decision is that they would pay for the whole trial, and which is very generous of them. Otherwise, we would not be here today. So what we did is that at that time, we started a proof of concept clinical trial, a phase 2 clinical trial. For those of you involved in clinical trial, you know that there are different stages. Phase 2 is really the intermediate stage, but this one is a good one. We managed to do double-blind randomized control trial. That means that the doctors are blind, they don't know what they're doing. The patients were not told whether they're taking a placebo or not. So we neutralized all those psychological effects of trial management. And we've been supported by Kidney Research UK, and most important among the audience, there's a person sitting here. On the day we designed the trial protocol, he sits among us, and he's a patient. And he tells us clearly what is useful for a patient, what is not useful. So the whole protocol we did at that time was really based on a lot of patients' input. So and then Kidney Research UK continued to support us in terms of patient engagement, which is very useful. And the trial is much harder than we think. At the end, we got to have 20 international centers collaborate together to make it happen. And we started in 2014. And we finished the last patient treatment in March this year. And so where we are now is that we are analyzing the data. But I got 1 or 2 slides to show you the preliminary analysis. But on whole, the trial, what we did is that we really have no confidence because it's the first time this class of medication using any patient with kidney condition. So we play safe. we thought the first thing, don't do any harm to your patients. And so what we did is that we give a short-term trial assessing the safety and how tolerable the medication is, and hopefully get some signal about efficacy. And we only treat the patient for 24 weeks, which is a very short time for a long-term condition. And the key inclusion criteria is that patient got to have a recent biopsy showing IgA nephropathy, features of inflammation, and then they need to still have more than half a gram of protein a day in the urine after their blood pressure is controlled, after they've been stabilized on angiotensin receptor blocker ACE inhibitor, because we don't want to expose the patient who got good kidney function after standard of care treatment to an unknown drug. And the key exclusion criteria is that there are some patients with scar kidney and very low kidney function. At that stage of the development, we thought it's not fair to ask them to join the trial yet. So that's a design of a trial. We try to have 3 groups of patients. At the end, what we achieve is 76 patients internationally, 25 on placebo, 26 on low dose, 25 on high dose. And all of them were given the medication orally. And this is the trial protocol. At that time, the main thing, the message take home is that initially, there's a lot of frequent visits in the beginning because we want to be sure of the safety of patients. But once they get used to the medication, then the visit become less frequent. And the key endpoint we're looking for is whether there's evidence of improvement of proteinuria, any improvement of histology, and the most important, how safe and how well tolerable the medication is. And so this is the distribution of the research center across the world. And UK got 6 hospitals and 20 of them across the world in total. And one of the things we're very pleased is that we managed to engage both North America and the Far East in this study as well. And this is the number of recruitment. You should be very pleased that UK done very well. So we've done 23 patients in UK, 23 in America. So consider America so much bigger. And we've done really well in recruitment. So the timeline is interesting. We first studied this molecule in 2012 in IgA nephropathy. So now we just finished treating the last patient in the trial. The question is, all of you want to know what the outcome is. I don't know the full answer to that because we have a lot of data we're analyzing at the moment. But what we think is that the patient who have milder disease, we're not sure we can demonstrate the benefit or not. So these are the patients who just got halfway between half a gram of protein in the urine a day after standard of care of treatment. There's a lot of baseline fluctuation of kidney proteinuria. The kidney function is very stable. And it's not easy to be sure whether our drug makes a difference. But now, when you look at the more advanced disease patient, these are the patients got more than a gram proteinuria a day, despite you do everything for them. Then we see something interesting. So there are some improvement on placebo. So you got about 15% reduction of proteinuria. Like all situations, for some strange reason, if you do a clinical trial on many conditions, the placebo group do have some benefit. And then what is interesting is that with the increasing of dose of SYK inhibitor, here you can see low dose, you got further improvement, high dose further improvement. But you notice that for all those professionals around here, I didn't have a p-value here because the statistic is not significant. What we see is just a trend of improvement. And I presume part of it is that when we look at the patient with more advanced disease, we get about 14 to 16 patients per group. So our sample size is too small. And there's no doubt that our treatment duration of 24 weeks is a very short term for a long-term condition. So the safety side is worth reporting. So on the whole, we know about the side effect of the medication. We didn't see any surprises. This drug is known to cause diarrhea and nausea and sometimes headache and hypertension. And high blood pressure is interesting because we knew this drug can cause high blood pressure, so we controlled for that, and patients were well treated. So we managed to neutralize that effect. And then in terms of how well tolerable, a small number of patients had adverse events, and one patient had a perforated peptic ulcer and died in a Far East center. And then we asked the independent investigator to look at that. They don't think it's related to the medication. But I think it's just fair to let you know that there's one patient gone through that. So in conclusion, I've shown you the data of our preliminary study where we go from kidney cell culture to international clinical trial. But the preliminary data show that patients more advanced proteinuria, they show some benefit to the drug treatment. But the sample size is small. We don't have statistical value to validate that yet. And then we're planning for further analysis and clinical study. And a lot of people help in this study, including, most important, the patient's input, not just being as a trial subject, but actually in design and also publicity of the trial. Thank you.